So when we're talking about the blood, we're talking about the liquid that will actually travel through the entire body through our cardiovascular system. The average volume of blood in the body will differ between male and female. This is going to be an adult male and adult female. So the average volume of blood in an adult male is going to be about 5 to 6 liter volume, whereas an adult female will be about 4 to 5 liters of liquid. Again, this will vary depending on height, stature, um, muscle concentration, but the average volume of blood for male and female are those numbers. Now remember from AMP1 that when we were talking about the blood, blood is categorized as a connective tissue. So in order to make it a connective tissue, it has to have two requirements. And those two criteria to be a connective tissue were to have your extracellular matrix and your fibers. So the extracellular matrix, when we're talking about the blood, is going to be the plasma. We're going to break this down in a second. And the fibers that make up this tissue is going to be the platelets. So as we start talking about plasma and platelets, these are the pieces that actually make blood a connective tissue. So first, we're going to break down what plasma is. So plasma is actually going to be 90% water. So this is going to be the bulk of the liquid in our blood. Apart from that 90% water, it is also going to be made up of nutrients because the blood is carrying nutrients throughout our entire system. It will be made up of gases. So it will be carrying oxygen, carbon dioxide, nitrogen throughout our system. Blood will also, or the plasma, sorry, will also carry our hormones. So like we talked about in exercise 27 with the endocrine system, all of the hormones have to travel throughout our body through this closed or open system, the cardiovascular system. So hormones are carried in the plasma part of blood. And then there will also be some wastes and metabolites, or the pieces of metabolic functioning that need to be removed from our system will be carried in the plasma part of blood. So plasma is awesome. It kind of makes up that criteria to be connective tissue. But when we're actually looking at blood, there are going to be two parts that make up whole blood. So whole blood is what you actually see if you were to take a vial of blood out of your system. Whole blood has two parts. The first part is going to be formed elements that make up blood. When we're talking about formed elements, I apologize, this is going to be made up of all the different cells that make up blood. We're going to talk about these next. So whole blood will have two parts. You have formed elements, which are going to be made up of a variety of different types of cells, and then you will have this plasma. So whole blood has formed elements and it will have plasma. So we've talked about what makes up for plasma. It's about 90% water. And now we're going to talk about what makes up those formed elements. So when we're talking about formed elements, we're talking about the cells. There are three main categories of cells that make up these formed elements. The first one is going to be your RBCs. This also stands for red blood cells. The technical term for red blood cells, or RBCs, is erythrocytes. So that will be spelled R-Y-T-H-R. Cyte. Cyte means cell. So red blood cells, another name for that, will be erythrocytes. These are going to be the molecule or the cell that will transport gases. The main gases is going to be O2 and CO2 in the body, or transport in the blood. So our red blood cells will transport our gases. They are also going to be made up of hemoglobin. That hemoglobin protein molecule in our red blood cells is what will allow it to transport those gases. We'll talk a little bit more about this as we go throughout this semester. So formed elements are made up of red blood cells. They are also made up of WBCs, also known as white blood cells. The other name for these are going to be leukocytes. Again, cyte is in the name. Leukocyte are our white blood cells, and these are going to be our defense mechanism. So they are involved with your immune response. And then the third formed element that will actually make up blood, or formed element that will make up this, is going to be our platelets. So this is where that fiber characteristic of having a connective tissue comes into play. Our platelets, these are going to be involved with clot formation. So when you need to have the production of a clot because there is a broken blood vessel, platelets will be the characteristic of our formed elements of our blood that will actually solve that problem. They are really, really, really important in maintaining homeostasis. So platelets are super tiny, but they are very important in being able to maintain our homeostatic levels throughout our body. So when we're actually looking at this whole blood, if you have ever gotten your blood drawn or you work in an environment, what they do is they take the blood out of the system, and this whole thing right here, when they take it out, this will be your whole blood. Inside this vial, when they take it out of your system, it will have your formed elements, which are all of these cells we just talked about. Also in this whole blood will be your plasma, which will be that mainly water-based, carrying nutrients, gases, and the other pieces we've already talked about. Your formed elements will make up about 45% of your whole blood, and your plasma will make up about 55% of your whole blood. If you've ever gone and seen what it looks like when they spin your blood down, we'll talk about this in a second. Essentially what they do is they are separating the formed elements from the plasma. So if they go through a process where they spin it down in a centrifuge, essentially what you get at the end of it is going to be a layer. And you will have this bottom layer will be full of your formed elements. So the solid will sink. And then it will separate and you have a top layer, and this will be all the liquid. So whole blood, when it comes out, this will all be red. This will be separated once you get your blood spun out. Okay, so next what we're going to do is we're going to take our formed elements, we're going to take our erythrocytes and our leukocytes and break down those characteristics. So we're going to start with our erythrocytes. So your erythrocytes, remember, are the red blood cells that you have in your body. These are usually about 7 micrometers in diameter, so they are circular. They're technically going to kind of have the shape that is called a biconcave disc. So they are circular, so they kind of look like this. And when we say biconcave disc, what we mean is that this middle section right here will actually be hollowed out. So if you were looking at it from the side, it will actually look more like an inner tube or a donut. So this would be the concave part. So I like to describe an erythrocyte as a Werther's original. If you've ever eaten one of those, that caramel candy or a inner tube that has the middle section still attached, it's a biconcave disc is what an erythrocyte looks like. Not necessarily a donut because it's not all the way through right here. It is just hollowed out in that middle. When you look at it underneath the microscope, you will have a pale center because the center of the erythrocyte is hollowed out. The outside areas will have its red coloration, and then since this section right here is a little bit more narrow, it will look pinker. So it looks like it's a red outside or red outside and then a slightly pink inside because of that pale center. These are the most numerous type of cell you have in your blood. So when you're making up our whole blood, your erythrocytes are the most numerous. They are also anucleated. So that means they are without a nucleus. And if they are anucleated, that means that they are unable to reproduce. They do not have the DNA in a nucleus because they don't have the nucleus to be able to replicate. So they are unable to reproduce. Because of this, they have a lifespan. And erythrocytes can only live for as long as their lifespan is, and it's about 100 to 120 days. So that means about every 120 days, your erythrocytes, all of your red blood cells, will have become turned over and they will all be new. So that's kind of an overview on our erythrocytes. Next, we're going to take our category of our leukocytes. And this is the bulk of today's lesson. So our leukocytes are our white blood cells. There are 5 types of leukocytes that we're going to talk about. So our leukocytes, these are our white blood cells. Some overviews of these guys before we get into the 5 different types of leukocytes that we have. These are all going to be nucleated. So that makes them different than our red blood cells. And in fact, the 5 different types, you can identify which ones they are based on the shape and size and color of the nucleus. But all leukocytes are nucleated. They are also found in our bone marrow. So not only are they found in our blood, but they're also found in our bone marrow where we can have that immune response. They are less numerous than our red blood cells. So when you're actually looking at our whole blood and our formed elements, there are more erythrocytes, there are less leukocytes. Okay, and remember there are 5 types. So we're going to break down these 5 types. These 5 types are broken into 2 categories. The first category will be granulocytes. And granulocytes have the identifying characteristic that they will have granules in the cell. So granules are essentially going to be a very small fragment of a cell. So if we were to have a cell like this, granules can be little tiny dots inside the cell, or granules can be large dots inside the cell, but there's granules inside the cell. There are 3 types of white blood cells that are granulocytes. The first one will be our neutrophils. Neutrophils is the first type of white blood cell that we're going to talk about. This is the most abundant white blood cell. So out of these 5 types of white blood cells, neutrophils are the most abundant. They make up about 50 to 70% of your white blood cell concentration. When we're looking at our neutrophils, we're going to look at the nucleus. The nucleus will have about 3 to 6 lobes to it. So we're going to draw this and show what it looks like. You're also going to look at the cytoplasm as a characteristic. The cytoplasm of a neutrophil will be pale lilac or light purple. And it is a granulocyte, so we have to talk about the granules inside the cell, and these are going to be fine, very fine granules. So based on what we know about a neutrophil, these are also going to be circular. Our nucleus is going to be about 3 to 6 lobes, so usually that's a 3-lobe nucleus, or sometimes you can see it as multi-lobed like that. But also what you'll sometimes see with neutrophils are this. You'll see a nucleus kind of look like this. But again, 3 to 6 lobes. So this has 1, 2, 3, 4, 5, 6 lobes to it. You're also going to look at the cytoplasm. And the cytoplasm is going to be a very pale color. to find a way to do this. So this cytoplasm, woo, not that one, that one, will be pale purple. And this cytoplasm will be all that color. And also remember that this is going to have granules. So this granule inside will be very, very fine, but it will have granules. So this is what a neutrophil looks like underneath the microscope. The next type of cell is going to be an eosinophil. Eosinophil. This is the second type of white blood cell. An eosinophil will actually make up about 2 to 4% of our white blood cells. So that is vastly different than our neutrophils taking up 50 to 70% of our white blood cells. An eosinophil is about 2 to 4%. His nucleus is a figure 8 or bilobed. So there will be 2 lobes to it, but it usually looks like a figure 8. It will have large granules in the cytoplasm. So very, very large granules in the cytoplasm. If we look at an eosinophil underneath the microscope, this is a lot of times what you will see. Where it's 2 lobes, it is a large figure 8 type nucleus. It will be dark, and then you will see large granules inside. Okay, the third type of granulocyte is going to be basophil. So we have our neutrophils, our eosinophils, and our basophils. Our basophils are going to be the least abundant white blood cells. So you do not have that many of these. In fact, you only usually have about less than 1% of your white blood cells being a basophil. The nucleus for these guys are going to be U or S shaped. So not to confuse that with an eosinophil, which is like a bilobed, but these are usually going to be a U or an S shape. They are going to be super, super large, coarse granules in the cell. And these are actually going to be very, very, very important for any type of immune response. So a basophil will come into play when there is the need for an immune response. It's part of its function. And it will actually contain histamine, which will help with immune response. So when you're looking at the basophil underneath the microscope, he looks like that. His nucleus will use, that's a U shape if you're looking at it. If you're going to do see an S-shaped nucleus on a basophil, he will sometimes look like that. They kind of look similar with the U and the S shape, and then you will have the very large granules in this cytoplasm of the cell. So those are our 3 granulocytes. You have a neutrophil, you have a eosinophil, and you have a basophil. So the next category is going to be agranulocytes. So these are white blood cells that are without granules in the cytoplasm. So we're not going to talk about what the granules look like with these. We're just going to talk about the nucleus or the cytoplasm as an identifying characteristic. So with agranulocytes, there's 2 types. First one is going to be a lymphocyte. So keep in mind a lymphocyte is a type of leukocyte, so don't get those confused. This is the smallest white blood cell, and it makes up about 25% of your white blood cells. So he's very small, but he actually makes up a decently large percentage. About a quarter of your white blood cells will be this type. When you're looking at a lymphocyte, the nucleus is going to be very dark. Usually about a dark purple is what you'll see. So it's very large and very dark. He's actually so large inside the cell that when you actually see the cytoplasm for this guy, it is going to be essentially just a thin line around the nucleus. So this nucleus is so large that he takes up a large chunk of space inside the cell. So here's our lymphocyte. What we're saying with our nucleus of our lymphocyte is he's going to be so large that he takes up this much space. And this is actually going to be a dark purple. Inside the cell. So this guy looks like this. He is dark purple. He takes almost up the entire section of the cell. But then you're going to have this little tiny section of cytoplasm around the outside of the cell that you will see. But you only see a sliver of this cytoplasm around the cell. There are no granulocytes because remember it is an agranulocyte, so we don't have to discuss having any granules inside. Okay, the next agranulocyte is going to be our last white blood cell, and this is a monocyte. A monocyte. So a monocyte is the largest white blood cell. Makes up about 3 to 8% of your white blood cell count concentration. And there is a abundant cytoplasm in these guys. So when you look at these underneath the microscope, you're going to see a large amount of cytoplasm and a dark nucleus. So when you see this monocyte underneath, it does say mono, so we're going to look for one essential lobe of this. So a monocyte looks like this, and I like to describe him as a kidney bean. So he kind of looks like this a little bit. But that inside nucleus will be a very dark purple. And then you will have the outside cytoplasm being that light color. So this is different. Keep in mind a monocyte is different than a eosinophil or a basophil, which is a bilobed structure that we talked about, because those are granulocytes, so they will also have granules. A monocyte may have a similar looking nucleus, but it does not have visible granules inside the cell. So those are our 5 white blood cells. When you're looking at these 5 white blood cells, there's a couple tricks to the trade that will kind of help you keep everything straight. So, okay, when we are talking about our white blood cells, they are usually going to be described in order of most abundant, so how many you have in whole blood, to least abundant, how little you have. So when you're doing your white blood cells, usually they're going to be categorized as most abundant to least abundant. Some tricks of the trade is never let monkeys eat bananas. NLMEB, from most numerous to least numerous. Never let monkeys eat bananas. So this would be our neutrophil, lymphocyte, monocyte, eosinophil, and a basophil. Never let monkeys eat bananas is how they are described. You'll also need to know largest size to smallest size, the actual structure of the cell. We are not really going to talk about all five. You just need to know the one that is the largest. That will be the monocyte, and the smallest will be the lymphocyte. So largest and then smallest. The other three kind of hang out in the middle size-wise. You just need to know the largest and the smallest. But the big thing here is never let monkeys eat bananas. Most numerous white blood cells to least numerous amount of white blood cells. Okay, so the next section we're going to talk about is some tests that will happen when you need to diagnose or look at levels of the blood. So these are going to be hematologic tests. All the different types of clinical assessments that can be done to look at the state of your blood. One of the first hematologic tests we're going to talk about are going to be something called a total white blood cell count. So total white blood cell count. So this is where you are looking at the total number of white blood cells. And you're doing it divided by total volume of blood. So since they can't take out the entire volume of blood, they normally just do one of those small vials, a couple milliliters. And what they do, in order to get the total number of white blood cells, they actually will count up all 5 types they see. So one of your exercises today is to draw a histology drawing, that is a blood smear, where you can see all of the 5 types of white blood cells and all of the erythrocytes. To do a total white blood cell count, you'd be counting up all 5 types of white blood cells you see divided by the volume of liquid. So that means some person is staring at a microscope and scanning and looking at all of these different types of cells. Some issues that why we need to look at your total white blood cell count, because there is a certain percentage of white blood cells that need to be found in your blood. If you are found to have an increased or a high white blood cell count, there are going to be some clinical issues that are different if you are found to have a low or a decreased white blood cell count. So if you are found to have a high white blood cell count, it is termed leukocytosis. Leukocytosis, because remember, leukocytes are white blood cells. So if you have an increased amount of them, you have leukocytosis. This is usually signifying some sort of infection. It could be bacterial, it could be viral, but for some reason your white blood cells needed to be reproduced and you have more of them, signifying some sort of infection. It could also be signifying some sort of disease, more of a metabolic type disease, an issue that your body itself has had to fight off. It also comes into play for diagnosing whether or not there's been poisoning, or if there has been hemorrhaging in the system. So leukocytosis is a high white blood cell count. This is going to be with numbers. The normal is going to be around, normal white blood cell count will be about 4,800 to 10,800 cells in a millimeter cubed volume of liquid. So if you're looking at essentially 1 milliliter of liquid, it's somewhere around 4,800 to 10,800 white blood cells. So a high white blood cell count would be somewhere that is above this number, and a low blood cell count would be below this number. So when you have a low white blood cell count, you're diagnosed with leukopenia. Leukopenia is where you are below 4,000 white blood cells per millimeter cubed of liquid. This is usually going to signify measles or typhoid disease. A lot of times this will come in when there is a diagnosis of cirrhosis of the liver, or when there is some sort of antibiotic use that is beyond what it should be. You could have leukopenia, where you've essentially depressed the white blood cell count, so you have a depressed immune system. Measles, typhoid, cirrhosis of the liver, and if there's antibiotic use. Okay, so that is white blood cells. There is another disorder that plays into white blood cells that we want to talk about, and that is going to be the disorder of leukemia. So leukemia, we've probably all heard about. If you look at the root word, it's leuke, which means it is a disorder of white blood cells, and this is actually going to be considered a malignant disorder. So this is where you will actually have uncontrolled proliferation of your white blood cells. So what that means is our white blood cells have the ability to reproduce because they have that nucleus that we've all talked about. Leukemia is an uncontrolled growth of the white blood cells that for some reason has no orientation of why it is needed. If you've heard about leukemia, sometimes called cancer of the blood, that essentially is saying that it is just like cancer in skin cells is going to be uncontrolled proliferation of your epithelial cells. Leukemia is uncontrolled proliferation of your white blood cells, and it is found in your blood. So blood cancer is the common term that's used for leukemia, but that is the science behind why it is called that. So technically it is an increase in white blood cells, but the reason why it's an increase is because there's just too much development of them. Okay, so just like you can do a total white blood cell count, you can also do a total red blood cell count. And this is going to be the same thing. It's the total number of erythrocytes that are counted up divided by whatever unit volume of blood is being used. So they're doing the same thing as the total white blood cell count. They're just adding up all of the red blood cells that they see. So if you have an increase in red blood cells, there are some clinical manifestations that will arise just like if you have a decrease in red blood cells. So normal falls right in the middle at about 4 to 6 million. So increased over that, you are going to be diagnosed with polycythemia. And if you are diagnosed with polycythemia, you have an increased level of red blood cells. This is usually going to be also a precursor to bone marrow cancer. So that would be something they'd be looking out for. And also, this can just, fun fact, also happen if you are living at high altitudes. So people in Denver may have a normal increased red blood cell count because they are living at a higher altitude, and so they have a decreased amount of oxygen in their system just by nature of where they live. Very interesting. If you have decreased red blood cells, we've probably all heard of this one. It's called anemia. So anemia, we usually hear about anemia in the context of iron, but if you have a decreased amount of red blood cells, you are essentially decreasing the ability to carry iron in the blood. So then you are diagnosed with anemia. So some interesting things about, we've all heard these terms, but this is kind of what they mean. Okay, so how they actually do these, one way they'll do it is to go back to the very, very beginning where I said, if you take a vial of blood that looks like this and the whole thing is liquid, and then you spin it down and you get that separation between the blood, that term is actually going to be called, you're doing a hematocrit. This is another hematological test called a hematocrit, and it is going to be packed cell volume. So you take this whole blood right here, it's got everything in it, all of your cells, all of your liquid, all of your gases. You put it in a centrifuge, you spin it down, you get this. And essentially what it is at the top will be all of the plasma, so it will be all of the liquid. And on the bottom, you will have all of the formed elements or all of the solids. And in the middle, right here, this is normally where you will get your white blood cells. So with a hematocrit, what they're going to look like is the reason the formed elements float to the bottom is because these are all of our cells. So if you actually take cells, they are made up of proteins. Proteins are heavy, so they will sink to the bottom, whereas plasma is mainly made up of liquid that is not as heavy, so that it floats to the top. But with your hematocrit, if you have this level of white blood cells in the middle versus someone who has this level of white blood cells in the middle, you can see the difference between how much there is here and how much there is here. This person would have some sort of infection where they have had an increase in their white blood cell counts to fight off whatever infection. So that's a first round of diagnosis that can happen in a clinical setting. Okay, so the last thing we're going to talk about in the world of blood is we've talked about the types of cells, what makes up blood, some blood tests, some hematologic tests. But the next section we're going to talk about is actually pretty interesting to us all, I'm sure, is how we get blood typing. What does it mean to have different types of blood? How do we determine that? What is that? So when we're talking about blood typing, what we are saying is it is some sort of system that has been created that is classifying blood. Sorry, y'all. So it is a system of blood classification. So we've taken blood and just tried to put it together and organize it to make sense. And the way we have classified blood is based on the presence of specific proteins. So your blood will have specific proteins. And these proteins are actually going to be found in two places. They are going to be proteins that are on the outer surface of your red blood cells. So blood typing is a system of classifying blood because we're saying that there is a presence of a specific protein on the outer surface of a red blood cell. To be even more specific, it's not just a protein, it is a glycoprotein, which means it's a sugar chain attached to a protein. So essentially it's a sticky protein. So our red blood cells, remember, are biconcave discs, so they look like this. When we're saying that there's a specific protein on the outside, we are saying that all the way around, actually, let's do it like this. All the way the outside of your cell, you are going to have some sort of specific protein that is attached to the outside that tells you this type of red blood cell is a star red blood cell. Okay, so these proteins, some things about these proteins. They are also going to have the name antigens. So what we're saying is that these are going to be antigens on the outside of a red blood cell. They are genetically determined. So that is how you can have the same blood type run within a family, is they are going to be genetically determined. Okay, every time you have an antigen on the outside of the body, you are also going to have something called an antigen. Oh, so sorry. You have an antigen on the outside, but what you're also going to have is a protein on the inside. And this protein on the inside is going to be called an antibody. So our two terms are antigen, and that is the protein on the outside of a cell. But also you're going to have a protein that is found on the inside of the cell that is called an antibody. And it's an antigen and an antibody that is what qualifies specific blood types. So the reason you have an antibody is because this antibody will react with red blood cells that carry a different antigen. So this antibody on the inside of the cell is what will react with blood that is a different antibody and different antigen on the outside of the cell. So the way we actually do blood typing is by a system called ABO typing. And the way this is done is it's going to be done by the antigen and the antibody. So we're going to build a little chart. Okay. ABO. We've all probably heard of these types of blood. So when you're doing blood typing with the ABO system, you are going to start with your blood type. Your blood type will have a specific antigen. And this is again going to be on the outside of the RB red blood cell membrane, but it is the antigen. And then every blood type will also have a specific antibody. And this antibody, remember, is going to be present in the red blood cell plasma. So the blood type will have an antigen and an antibody. So our 4 types of blood are going to be A, B, AB, and O. And these are named essentially for the antigen. So blood type A will have antigen A on the outside of the cell. So we're going to have A, A, A. So blood type B ooh, there we go. Blood type B is going to be named the same way, off of the antigen that it has. There we go. So blood type B. So this blood type will have the proteins B all the way around the outside. Antigen or blood type AB will have antigen A plus antigen B. So on the outside of your cell, he will have some A's, but then he will also have some B's. So he has both proteins on the outside of the cell. Whereas if you have blood type O, you actually have no antigens. So you just have your red blood cell. So essentially it's a 0. You have none. So every blood type also has to have an antibody. So blood type A, which has antigen A as the protein on the plasma membrane, will have antibody that is the opposite, which will be anti-B inside the plasma membrane. B blood type, which has antigen protein B on the plasma membrane, will have an antibody inside that is an anti-A antibody. So if we're going to do anti-B as little plus signs, we're going to do anti-A as little minus signs, and that's what they have on the inside of their plasma. Blood type AB, which has antigen proteins A and antigen protein B on the plasma membrane, will have no antibodies on the inside. So it will not have any proteins on the inside. Whereas blood type O, which has no antigens on the plasma membrane, will have both antigen A, anti-B, and anti-A inside the plasma membrane. So it will have both anti-B and anti-A on the inside of the plasma membrane. So these 4 reasons are how you get the blood type A, B, AB, and O from a protein called the antigen that is on the plasma membrane of a red blood cell and from an antibody that is inside the plasma of a red blood cell. So this is how you will get that. So that kind of wraps up our blood system for exercise 29.